I actually started out as a general practitioner. Can you hear me in the back? No? Okay. I actually started out as a general practitioner, and this past summer I retired from Cornell University. And I've specialized in pathology, and I also have certification in American College of Sports Medicine. But for more than a decade, I've been researching and intrigued by the causes of type 2 diabetes. I'd like to share with you some today and explore some of the insights into the paradox that this question presents. How can a disease that runs in families not be in our DNA? So when you go to the doctor and you fill out one of those intake forms, invariably one of the questions is, do you have diabetes? And for 1 out of 9 Americans, the answer there is yes. And then another question is, do you have prediabetes? And for 2 out of 5 Americans, the answer again is yes. So if you add that up, those two numbers, you get 50%, a little over 50%. The chances of answering the question is, does it run in your family? Are the genes for type 2 diabetes inherited? Are they in our DNA? If our parentage is to blame, isn't it a lost cause, especially if we're diagnosed with prediabetes? For if it's in our DNA, how is there any room left for us to effect an escape from our destiny? So, in other words, if that plane of between blameless victim and personal responsibility goes to the right, then there isn't that much room left. Now, 42 years ago, in my first medical school lecture about type 2 diabetes, the lecturer told us about the Pima Indians. The Pima Indians who are in southern central Arizona. They're said to be genetically predisposed to type 2 diabetes. And in fact, astonishingly and tragically, more than 50% of the adults have type 2 diabetes. I've actually fact-checked him, and the percentages are correct, but the details are revealing. For thousands of years or more, the Pimas grew beans, squash, corn alongside a flowing river, the Gila River. And in the late 1800s, settlers to the north diverted the flow of the Gila River. And with that, death and famine regularly visited the Pimas as the waters ebbed and gradually receded. The U.S. government stepped in, providing a long-term diet with rations, which could be preserved for a long time. And that's key. The Pima's plant-based, low-fat diet gave way to a high-fat, low-fiber, highly processed diet. And without fields to hoe and weed, they became less active. Now, one last crucial detail my professor failed to mention is that the Pimas, right across the border, they have a Mexican cousin tribe. They're also called Pimas. And these Mexican Pimas have a prevalence of type 2 diabetes, which is 1/4 of their American brethren. So what was the prevalence of type 2 diabetes 150 years ago? Now, nobody knows for sure, but I tried to guess and doing a back of the envelope calculation. Any of you recognize this name, Elliot Jocelyn? Elliot Jocelyn's a doctor. He specialized in type 2 diabetes, and he wrote extensively. So he was saying, why should the incidence of diabetes double in the U.S. in the 10 years from 1880 to 1890, and then take another 30 years to do so between 1900 and 1930? And he further wrote that type 2 diabetes was a rare disease compared to type 1 diabetes. That's a shock for many of us. Now, doing the back of the envelope calculation, because Dr. Wilkerson in Oxford, Massachusetts, did actually a prevalence study. And he came up with type 1, type 2 combined was 1.7% of the population at the time he did this study. So I took the two numbers. Now, you have to give me a little leeway. It's a little sloppy math, but that comes to a type 2 diabetes prevalence of 0.4%. Now, if you remember earlier, I said it's 1 out of 9 Americans. So if you do the math, it's not a doubling. It's not a tripling. It's not a quintupling, or even 10 times. That's 27 times the increase of 150 years ago to the present day. And in 150 years, you do not get that kind of 27 times rise in a genetic disease. So modern studies have looked at the genes which are associated with type 2 diabetes, and they're listed here. These genes control these various parts of our genome. But if you did a 23andMe test and you found that you were positive for some of these genes, it still wouldn't predict whether you had type 2 diabetes. You could be positive for 23andMe and not have diabetes or even prediabetes. The reason is that these genes are widespread in our population. They're the same genes that were useful to our ancestors. They helped us survive lean times and fatten up for those times leading up to that. So everyone here, sitting here in the audience, is an offspring of someone who carried these genes because they were useful to us. Now, I should confess, I started this lecture and I served you up a pair of false choices. Something in logic which is known as a false dichotomy. A false dichotomy, the premise is wrong. There may not be only two choices. And certain diseases, even tumors, are caused by single diseases on the left side here. These are single gene diseases. We're familiar with those. And for example, retinoblastoma is a type of tumor of the eye that if you have the gene, then you'll get the tumor. And on the right side, you'll see these environmental diseases. And you can see this, skin cancers due to sun exposure, tuberculosis, exposure to the bacteria, smoking, emphysema, lead poisoning. These are things which are due to exposure to the environment. But there's a middle category, and this middle category is called epigenetic diseases. Scientists have come to realize that certain groups of genes can be turned on by environmental triggers, lifestyles, and diets, because this epigenetics allows us to tune our DNA to our environment. So we'll talk a little bit more about that. This center category, this middle category, is vast. It accounts for 3/4 of the diseases of our modern society. So if you go into a doctor's office and you look around, 4 people, 3 of them have epigenetic diseases. So whether a disease, especially an epigenetic disease, runs in a family, it has more to do with that family's lifestyle than their shared DNA. So we have the ability to modify the expression of our DNA, but not change it. And how we live is the key to turning on or turning off our DNA. Now you can think of epigenetics like a fly fisherman. Now a fly fisherman, this guy, he's got this pickup truck and he's got tons of gear. In fact, he's got gear not just from his father, from his granddad. He never throws anything away. This is like our DNA. We've got a ton of DNA which we've never thrown away. And the problem is that when he goes to this stream, he's got to figure out what to bring to the stream because his waste pouch there is only so big. And so it depends on the location, the season, the weather, the local conditions. He has to decide what lures or sinkers or flies to bring. With epigenetic disease, we pretty much have the same situation. We have to select from our massive DNA library about what to bring. And that depends upon what we experience, what we eat, and how we exert ourselves, and the environment that we immerse ourselves in. So, for those who don't care that much about how this works, indulge me for the next 2 slides. But I want to show you how this works. This is the DNA. This is, and it's unfurled and unfurled and unfurled. And then down here, the DNA is wound up around these spools here. These spools are called histones. And you see that little red segment in there. That red segment, even though it's part of our DNA, it's not expressed because it's tightly wound. It's only until something from the environment, whether it's exercise or it's pollution or some kind of influence that comes to bear and attaches to that histone, that it becomes unfurled, and then that DNA becomes expressed. So if we look at it closer up, we can see it better. So this DNA is tightly wound. It doesn't matter if it's in our library or not. It gets unfurled, and then it gets expressed. Now, I want to take a little sidetrack, and this is, I think it's fascinating to think about this. Why do we even have cancer? Why do the genes for cancer exist in our library? And this is a teaser. Actually, I've asked physicians this. A lot of them don't know. Only turns out pathologists and oncologists know this answer. It's because it's the price we pay for propagating the species. These are the same genes that help the baby impregnate itself on the uterus lining, invade the uterus lining, recruit blood vessels, and the cells from within the baby to migrate to their locations. The same type of genes that are in play in cancer. Except in cancer, what happens is they are activated when we don't want them to be. So, how long does epigenetics span? Up here, I've got a picture of your maternal grandmother. This is your maternal grandmother, and this is your mother. And see that G3? That's you. So this is about 3-4 months into the gestation. And this is your grandmother, and what she experienced leading up to that pregnancy and during that early pregnancy affected your mother. No surprise there. We all know prenatal care is important. But see, in your mother, the ovaries were forming, and those eggs were forming, and then at a little before the birth of your mother, those eggs were frozen in time, awaiting the sperm of your father to make you. So that environmental influence of your maternal grandmother and your mother in turn affected you. Now, all these downstream effects, they do have lessening effects as it gets closer in time to you. But actually, you have the biggest influence of what gets activated and doesn't get activated. It depends on how you live your life. So our DNA library has the genes to deal with starvation. It knows what to do with exercise. All these things it's seen before. It knows what to do with these triggers. Now, the problem that we encounter is when the DNA encounters a situation it's never seen before. Because, you ever watch MacGyver? MacGyver, he always cobbles together stuff this Cobb job in the show and he saves the day, right? But will that solution last into the following week, next month? No. And that's what typically happens, is we have a situation in which we haven't seen a situation before, and it gets pieced together, and it might work initially, but more likely it's going to end up in a situation that we don't want. So what type of situation is that? That situation is the last 150 years. So this is the slide I made up. It's got 4 different categories, just kind of arbitrary: technology, hygiene, health, lifestyle, and food. So all these 4 categories, you can take a look, think about yourself, your mother, your great-grandmother, and when were each of those in time when they experienced the end of that. And I'll give you an example. I think that most everybody sitting in this room were the last generation to experience no air conditioning, right? What about no refrigerator? Right. So each one of these things in the last 150 years, whether you consider them good or bad, they affected our genome, or epigenetics, as it were. Now, I want to focus on the last column here on the right. Now, all these actually do affect the expression of type 2 diabetes, but let's just talk about this last column for today. We have an unusual situation, and that is bumper crops. We live in an era unprecedented. We have an abundance of food. If you look at this black line right here, this is the yield per acre. We've gone from 20 to 100. That's a 5 times increase. We've experienced an unprecedented increase in food production. In fact, we have a situation here. This is a busy slide, but I only want you to take away one point here. See this point here, edible food? This is our edible consumed food here. See that exports? We produce twice as much food as we can eat. In fact, the number one thing that's going in our landfills is food. And so when I say we have a problem, it's actually not true. It's the food industry which has a problem, because they're competing for every one of your mouths. And there's only so much of that. So they're trying to get you to eat their brand of food. And that's the dilemma they face. Let me use this example. This is Cornell University, and this is Stocking Hall, and this is where the food science building is. Now you may say, food science, isn't that the same as nutrition? It's not. Cornell actually has the School of Nutrition and the School of Food Science. Now you would say, what could possibly be the difference between those 2 degrees? And for the first 2 years, they're actually the same. They study, as you would expect, chemistry, biology, those sort of things. But after 2 years, the nutritionist graduates with a degree that's focused on nutritional value of food and the impact on health, just like we would expect. But what about the food scientist? The food scientist studies preservation of food, marketing of food products, and how to process them. Where do you think these people go to work? They work in the food processing industry. So these food scientists, they work with the psychology graduates who are well-schooled in addiction, and they work with marketeers to make food that you will try to maybe resist. Maybe you want to eat less processed food. Maybe you want to exercise more, or try things which are non-drug related, except try to figure out which one of them is you and which one of them is them. It's not even, because they have got it honed down. These are snacks you recognize, and these are the slogans. I actually went and matched them up with the products, and I would take them at their word. These are addictive. And how are these made? These are corn, soybeans, wheat, and it's dried, it's pulverized, it's made into shapes, stamped into shapes that fit into your mouth because they want you to mindlessly eat as many as possible. And at that stage, actually, you would find it quite tasteless. It would say, oh, this tastes like nothing because it's not fresh. It's preserved just like the food the pumas ate. Except this industry knows how to add back flavors. They're artificial flavors. They're artificial sweeteners. And just like the pumas, they have to get it to the store, and it has to sit on the shelf for a long period of time, because otherwise, if it's spoiled quickly, the profits would go down. You can't make these foods in your kitchen. These are ultra-processed foods because they have ingredients that you don't have in your kitchen. In fact, if you look at those ingredients, polysorbate 80, it doesn't sound at all like food because it's not food. These are food additives. Now, these ultra-processed foods have unnatural additives. Some of them are out and out bad. For example, trans fats. Trans fats, are now banned in the U.S. with some exception. When they passed the law, they made some exceptions for, commercially baked goods. But trans fats are the result of ultra-processing. And the body doesn't know what to do with it. It's that MacGyver problem. And it causes atherosclerosis. And that's why it was banned. We knew that for decades. Why it took so long, I have no idea. Now, what about high fructose corn syrup? High fructose corn syrup is liquid. It's more than twice as sweet as glucose. And, in diabetes research, we need an animal to test diabetes with. How do we make diabetic animals? We feed them high fructose corn syrup. So high fructose corn syrup in high doses causes diabetes. Now, what about the third category? Unknown and untested. Unknown and untested is the great majority of additives, because there's in the law that governs these substances, there is a major loophole that says if the company submits an application and says, we tested this and it's safe, then it's accepted by the government. So what are in these? Emulsifiers and preservatives. So for example, nitrites. Nitrites, you see them in processed meat, deli meat. Nitrites, when they hit the stomach and in an acidic environment, they turn into carcinogens, or class 1 carcinogens. Preservatives, emulsifiers, those are emulsifiers, for example. Those are the ones when you get peanut butter, it doesn't separate. It maintains a layer. That's why the organic peanut butters, you have to stir them. That's a natural state of water versus oil. Now, what does that do? Well, the membrane of cells is fat, and they try to keep out water. So basic research tells us it's disruptive to bacteria cell walls. So emulsifiers have been implicated in disrupting our gut microbiome. So that's one theoretical reason why we can to avoid these. But I want to focus on this question here. No one prior to the last century, 1900s, sat down at the dinner table and said, I'm only going to eat 2,200 calories, right? No one did that. And yet they had no problem with obesity. How could that be? It's because we have internal mechanisms that control how much food we eat. Now, I want to just briefly go through this with you. When we eat, food comes down and it stretches the stomach, and then we have this ghrelin. Now, ghrelin, you might have heard, that tells the body I'm hungry. And then the food moves from the stomach to the small intestines. And the first thing that's removed in processed food is fiber, because fiber affects the storage ability of food. So almost all processed food is low in fiber. Now, it goes down here, and ultra-processed food is usually absorbed in the first third of the intestines. Now, what happens when you have low fiber and it's mostly absorbed? Okay, GLP-1, everyone heard of GLP-1? GLP-1 are those whiz-bang drugs for obesity: Ozempic, Mounjaro, Zepbound. The small intestines releases GLP-1. In fact, it's mostly the second half of the intestines that produces GLP-1. Guess what? GLP-1 is low in those who are overweight and those who have type 2 diabetes. It's kind of like an enhancer of insulin. Now, insulin and leptin form the four, what we call quartet. Quartet, they control the feeling of satiety. So when they get together and they're in balance, they tell the brain, hey, stop eating, we're getting full. Okay, now what happens in ultra-processed foods? In ultra-processed foods, you have artificial flavors and artificial sweeteners. So the body drinks this, eats this, and says, this must have calories, for example. Let's, for example, take the zero-calorie drinks. And it says, this must have calories. But it takes it, and there's no calories. So that kind of confuses the body. And then it has a high caloric density. In other words, for the amount of food, calories in a particular food, such as like a carrot or potato, for example, high caloric density foods have a lot more calories. So it doesn't stretch the stomach as much. And sure enough, people who have obesity or type 2 diabetes, they have a problem with ghrelin. That's not at the normal level. And these preservatives, emulsifiers we talked about, they affect our gut microbiome. And so GLP-1 levels are not normal either in people with obesity or type 2 diabetes. In fact, neither is the level of insulin or leptin. All four of these hormones, which control how much food we eat, are all off in the wrong direction. So how much do you need to be off? You only need to be off about 150 calories a day. So pharmaceutical companies to the rescue. GLP-1. Now you could increase your GLP-1 level by eating foods with more fiber, more natural fiber. Okay? These GLP-1 drugs increase the bloodstream levels 10 times of what they are physiologic. But one thing strange happened after we realized that they help and reduce weight, is that we realized they're actually psychoactive drugs because they mainly work in the brain. They work in the part of the brain that's the same as cocaine addiction, opiate addiction, smoking, you name it. How do we know that? It's because people who take these drugs have an easier time stop smoking, drinking alcohol, and getting off opiates. So actually there's a lot of talk in the medical circles because everyone's very excited that we're going to expand the use of GLP-1 drugs. In fact, the problem is 1 out of 3 stop GLP-1 drugs after 1 year. Now there's lots of reasons. One of them is cost, but the other one, which is strange, is that they lose the joy of eating. They lose the joy of eating, which is very interesting because you think about it. What is it that someone who uses drugs makes them want to keep using drugs? It's because it makes life a lot more interesting. In fact, regular life after cocaine doesn't seem quite as exciting. And that's what's happening with ultra-processed food, is that these food companies have concocted these substances which are more exciting to eat than normal food. So a successful diet will break a compulsive habit link. Now, I have a suggestion for you, actually. We're almost toward the end of the talk. If you don't think this makes any sense, go the other way. 80% of our children eat an ultra-processed food diet. 50%, according to Johns Hopkins, a few weeks ago came out a study, we adults in the US eat, on average, 50% ultra-processed diet. That works out to 17 pounds of additives a year. So go the other way. Instead of fight it, eat everything ultra-processed. Don't eat anything that's natural, and see how you feel. If it makes you feel okay, well then that's all be amazing. Please take a card and talk to me. But I'm willing to guess you will not feel very good. So how do you break this habit? So first thing is try to cut down on ultra-processed food. Don't buy them, obviously. Now, this is something really I thought intriguing. How many of you heard of something called the potato hack? Now, I was a general practitioner for the Air Force, and this guy in the Air Force came up with it. He was a master sergeant. He lives in the North Pole. He's retired from the Air Force. He had trouble losing weight, and so he decided, I'm going to research it, and it came out. This was actually done before in the 1800s, this idea of eating only potatoes. So under this diet, which lasts for about a week, maybe 10 days, you can eat baked potatoes, mashed potatoes, boiled potatoes, put a little salt on it, maybe a little oil to cook it, but that's it. That's it. You can eat it with the skin. And what do you think happens? Well, you lose weight. You lose weight because first of all, it's kind of boring. And second of all, you increase your fiber intake. Don't eat mindlessly. Okay? You know, in Vegas, you go to Vegas recently, they replaced those one-armed bandits. And the reason they replaced them is because they found out you can push a button fast and you can pull the handle. So ultra-processed food are designed for you to literally stuff your face. Okay, so this is my last slide. Ultra-processed foods are not food. They're not food at all. They are addictive and they have potentially damaging additives. We have diseases like type 2 diabetes because we're ill-adapted to live in the present era that we find ourselves. Our genes that we have for type 2 diabetes are present in all of us, but they're confused by what they're seeing when they come into our body. Unhealthy lifestyles, including ingesting ultra-processed foods, may activate these genes and lead to type 2 diabetes. So I put this up here. I'm giving this talk one more time. This is the Healthy Cooking Club in February, and I teach a course through the Enrichment Academy about lowering glucose without drugs. So I think at this point I'm going to turn it from a monologue to a dialogue. So thank you.
Thank you, Bill.